Today, we'll explore the fascinating world of mass spectrometry technology. But first, let's define mass spectrometry. It is an essential analytical technique used to identify and quantify a specific target. Mass spectrometry technology enables a wide range of applications in various fields, such as medical testing for protein analysis, drug metabolism, drug detection, biomarker detection, genomics analysis, virus and bacterial detection, and more. In mass spectrometry, a sample is subjected to ionization, acceleration, separation, detection, and data analysis. By examining the ion charge to mass ratio and relative abundance, the technology can determine the molecular structure and composition of the sample. This powerful tool has numerous applications in chemistry, pharmacy, biology, food, and environmental detection. For instance, in chemistry, mass spectrometry can be used to analyze chemical composition, investigate the ionization and fragmentation processes of reactants, and optimize material properties. Now let's discuss the different types of mass spectrometers available. According to varying needs and sample states, there are many distinct types of mass spectrometers. For our purposes, we will focus on liquid chromatography triple quadrupole mass spectrometer, which is the most common. Let's dive into the world of liquid phase and understand how liquid chromatography mass spectrometry, or LC-MS, is used to analyze blood samples, using prostate-specific antigen, or PSA, as an example. First is sample preparation. To begin, the collected blood sample is centrifuged in a centrifuge tube. After this process, the blood sample is separated into serum and blood cells. Next, a PSA extraction reagent is mixed with the serum, and the precipitate is removed using methods such as centrifugation and ultrafiltration. Second is liquid chromatography separation. The prepared sample is then loaded onto a reverse phase chromatography column where it enters the column. This column can be made of stainless steel, glass, or plastic and has a hydrophobic stationary phase and a hydrophilic mobile phase. In the column, nonpolar compounds linger while polar compounds are quickly washed out. By monitoring the absorbance or fluorescence signal at the reverse phase chromatography column's outlet, the elution time of the target molecule can be determined, allowing for the differentiation of various components. As PSA is a protein and a polar compound, reverse phase chromatography effectively separates and enriches PSA in the serum. Third is mass spectrometry detection. After the PSA molecules have been separated, they are directed to the mass spectrometer. The sample is sprayed into the ion source using an electrospray needle, transforming it into tiny droplets. Under high voltage, these droplets become highly charged. Only ions with the appropriate mass-to-charge ratio, or MZ, can enter the mass spectrometer. The ions are then controlled by a magnetic field and accelerated to high speeds, colliding with the glass molecules in the collision cell and fragmenting into smaller pieces. The second quadrupole, Q2, filters specific ion fragments based on their mass-to-charge ratio and sends them back to the collision cell for further fragmentation. Finally, the third quadrupole, Q3, selects the ion fragments of interest and guides them to the detector for analysis and recording. This entire process allows for the accurate identification and quantification of the PSA molecules in the sample. Fourth is data analysis. After obtaining mass spectrometry data, software is employed to calculate the concentration of PSA. Mass spectra are graphical representations that typically plot mass-to-charge ratio on the x-axis and ion signal intensity on the y-axis, resulting in peaks on the graph. The position and intensity of each peak indicate the mass and relative abundance of an ion. For example, in the simple case of water analysis, the integer mass of water is calculated as 2 times 1 plus 16 times 1 to 18, and the peak at MZ18 represents water. Similarly, Other substances can be detected based on their MZ values. By analyzing and interpreting the mass spectrum, it is possible to determine the presence, relative abundance, and structural information of different compounds in the sample. For example, the graph above shows that the PSA concentration in the blood sample exceeds 75 nanograms per milliliter, with a corresponding increase in PSA dimer and an alpha-1 ACT concentration exceeding 2 nanograms per milliliter, indicating a significant risk of prostate cancer. Liquid chromatography mass spectrometry technology offers several advantages for sample analysis, such as high sensitivity, high resolution, and high accuracy. The liquid chromatography triple quadrupole mass spectrometer can detect various non-volatile compounds beyond PSA, including proteins, peptides, nucleic acids, drugs, and metabolites. Another widely used instrument is the gas chromatography mass spectrometer, which detects gases and volatile compounds such as organic compounds in gas, urine, blood, and other biological fluids. Additionally, the atomic fluorescence spectrometer, or the AFS, is employed to measure the content of metal and some non-metal elements including copper, iron, zinc, selenium, and lead. In summary, the entire process of analyzing mass spectrometry data involves calculating the mass concentration of the target molecule, in this case PSA. The mass spectra, with their peaks representing various ions, provide essential information about the presence, relative abundance, and structure of different compounds in the sample. This information can be crucial in determining, for example, the risk of prostate cancer. Liquid chromatography mass spectrometry technology, with its numerous advantages and applications, has become an indispensable tool in various scientific and medical fields.